Our first speaker is Keerthi Krishnan. She's an assistant professor at the University of Tennessee, Knoxville. We'll put her lab website and Twitter handle in chat in a minute if you'd like to learn more about her work. So Keerthi, please take it away.
Great, thank you. Are you able to see my screen now?
Yes, great.
All right, okay, so thank you for the opportunity and for the introduction. All of our insights related to this title are due to our work on Rett syndrome. The next two speakers in this session will also be discussing Rett syndrome and mouse models in general. So I'm going to start with that as an introduction. Rett syndrome is diagnosed as a neurodevelopmental disorder that affects 1 in 10,000 girls and women worldwide. Many girls with Rett syndrome exhibit typical, normal, early development and then regress with atypical speech, gross, and fine motor issues. The hallmark of this disorder is this hand-wringing phenotype that you see over here. Many Rett syndrome patients live well into their middle age. It's currently unknown how the syndromic trajectory changes as the girls go through these physiological changes, such as puberty and menopause. Due to these studies in animal models for this disorder over the past 2 decades, from many groups, including ours, detection and diagnosis of early sensory processing is being used as a clinical diagnosis from now. Mutations in MECP2, an X-linked gene, causes Rett syndrome. It is predominantly found in the nucleus in this beautiful punctate pattern that you see here in green. It is found in most cell types, including our favorite, the parvalbumin positive GABAergic neurons, shown here in blue, which are surrounded by these extracellular matrix structures called perineural nets, shown here in red. Ms. Dana Leo, our next speaker, will be talking about these structures next. MECP2 is thought to be a master gene regulator that could affect chromatin compaction and or affect gene expression by associating with repressor or activator compounds. The community as a whole is trying to work on this puzzle of understanding how MECP2 function could relate to the Rett syndrome pathogenesis and symptomology throughout life. So one hypothesis for this connection is this presented here. Based on the studies from many groups, including ours, MECP2 could be considered as an epigenetic regulator of critical period plasticity, not just during early development, but perhaps into adulthood. This is illustrated nicely in this graph over here, where we see the expression of MECP2 increase, which is shown here in red, in relation to the known distinct critical periods for sensory processing, such as hearing, vision, and motor learning. Here, I'll focus on this hypothesis by introducing maternal behavior as a paradigm to study adult experience-dependent plasticity in the female heterozygous mouse model. Parental behavior is a great model to study adult experience-dependent plasticity, as the early developmental critical periods have long been established. With pregnancy and the need to take care of the young, there are new demands on these already established circuitry and established behavioral patterns. We use the alloparental behavior as a model here, where parental care is provided by other females besides the dam in order to focus on adult experience-dependent plasticity mechanisms over changes induced by the pregnancy hormones. Here's a schema depicting our alloparental behavior paradigm. A pregnant female is co-housed with 2 naive virgin females, a wild type and an MECP2 heterozygous female. These are later mates. 3 to 5 days prior to the birth of the pups. From the day the pups are born, which is day 0, till day 5, we perform the pup retrieval assay. During the actual retrieval assay, we start by removing the 2 other adults and any remaining extra pups out of the home cage and habituate the 1 adult and the pups for this particular assay for about 5 minutes in the home cage with the recording setup. Then we remove the pups and prime the adult in the isolation stage. Next, we scatter the pups in the different corners of the cage to initiate this retrieval process. After about 10 minutes of retrieval and recording, we start the process again for the other adults. After day 5, we go through and perfuse the wild type and the heterozygous surrogates. extract the brains, and perform immunostaining and microscopy for PNN structural analysis, which you'll hear about a lot afterwards. So mice perform this alloparental care of the pups very well. Here's a video showing that assay. In this, a wild-type female is retrieving the pups that are shown in the different corners here. This is done in a dark box. I'll let you watch for a minute. You can see that she's pretty good at retrieving the pups, finding them and retrieving them to their nest. These are not her own pups. And here's the MeCP2 surrogate het, the heterozygous female. Let you watch for a second. As you can see very clearly, the het goes from pup to pup, but doesn't actually retrieve the pups to the nest. Okay. From a Rett syndrome perspective, this is a really nice assay to look at primary sensory processing deficits, but also multisensory integration, which is important for social communication and higher order cognition. Okay, when we quantify these behaviors, which was published in this work in 2017, we look at latency and errors. Latency calculates the time it takes each adult to retrieve the pups back to the nest. Each circle here is an average latency of an individual mouse over the 3 different days. Though there is significant variability between the mothers and the surrogate wild type, it's clear that the surrogate het has significantly worse performance in this index. And now when we look at the errors, which are metrics for where the pups are being interacted by the mother or surrogate wild type or the het, but then actually not retrieved back to the nest. Again, the 2 other mothers and the surrogate wild type do show some variability in this, but the surrogate het has a significantly worse performance in this metric as well. So in the rest of this paper here, we focused on the auditory component, and I suspect that the last speaker will be talking a lot more about this later on. So when I started my own lab at the University of Tennessee at Knoxville, I decided to follow up on these error metrics and focus on this proximal tactile contributions. The idea was that they are able to find the pups and interact with the pups, but they're not actually bringing them back into the nest. But before we needed to focus on that, we needed to actually make sure that the assay works at the University of Tennessee. And so we decided to redo these assays here with a minor change where we did the behaviors on every single day rather than on day 0, day 3, and day 5, as was published previously. So what we see, first of all, is that the surrogate het do perform worse compared to the wild type animals. So reproducibility crisis has been averted. But we do see this nice behavioral rescue in some areas from day 2 to day 4, which then gets worse again on day 5. This is something that we noticed previously, but we didn't actually hit significance in those data analysis, but perhaps doing this assay on every single day is making this a little bit more clearer for us. Due to these dynamic changes in the latency and the errors, and also that the surrogate wild type is actually getting better throughout these days, we decided to do a systematic frame-by-frame analysis of the retrieval behavior using DataVyu software, which links videos to an extensive coding spreadsheet. The coding was performed by a tenacious undergraduate student, Ms. Parker Stevenson, in my lab, with extensive guidance and mentorship by Dr. Devin Kassenheiser, our collaborator at the University of Tennessee Health Sciences Center. Devin also performed all the statistical analysis from this coding information. I'm going to focus on 2 key data visualizations here. We plotted the sequences of these goal-related movements. We identified about 9 of them, which seem to be very important for this process. The surrogate wild type is shown here on the top. This is for day 0 of retrieval, and surrogate het over here. These are individual animals performing the behaviors, and the x-axis here is the time. So it's about 10 minutes of the retrieval time, and each block here is about an 800 millisecond time frame. So what we can immediately see is that each block, each behavior actually doesn't look the same as another one. So there's a lot of individual variability across different animals. The second part is that the gray, which is the search phenotype, and the retrieval, the full good retrieval, is the pink phenotype. You start seeing those emerge in the surrogate wild type pretty well. Whereas in the surrogate het, as we had previously shown, they don't actually do that process very well. The other parts that we notice is that there's a lot of yellow, which is the pup approach phenotype, and light blue, which is nesting phenotype. So still remember that the pups are scattered in the different corners. They're not actually in the nest. This is the hets that are hanging out in the nets. And there's also abnormal behavior shown here in black, which is mostly jumping phenotype. When we look at these behaviors on day 3 and day 5, we notice that the surrogate het actually improves. Many of them improve in this behavior, but by day 5, they actually get worse again. The abnormal behaviors are not as much as it was on day 0, but you can still see the pup approach, nesting phenotypes, and grooming phenotypes emerge in these hets on day 5. The wild types by now have completely crystallized this behavior, where you only see the gray and the pink, which is the searching and the good retrieval process. Then we calculated transition probabilities to determine what the probability of seeing a particular goal-related movement was, given that the adult was in a search mode. The main takeaway here is that the big arrows and the thicker lines that you see are the higher probabilities. And you can see that the surrogate wild type is crystallizing this behavior by seeing this gray to the pink transition quite well, with lesser arrows going towards these other 9 behaviors. Whereas the surrogate het, as you might have expected, has arrows going all over, lines going all over, suggesting that the transition probability between these different behaviors are quite variable in these animals. Okay, so in conclusion, it's important to take the individual behavioral responses over time into account. Individual animals, definitely, but per day is also very important for this. And according to the sequence analysis, we would actually say that's true for time as well during the assay. Tactile stimulation by whiskers, face, and somatosensation is very important for this behavior. This has been shown in wild-type animals, mostly rats, in the 1960s and 1990s. And we've also started performing some deep lab cut analysis so that we could start taking these assays with this gold standard that we have from Parker's data set to now start looking at supervised machine learning approaches for automating this purpose further. This is a study example here by Mike Mikins, a graduate student in the lab. So in addition to the people I've already mentioned in my talk, none of this work would be possible without my partner at home and at work, Dr. Billy Lau, shown here, and the rest of the Interpret undergraduates in the lab. I'm happy to take any questions. Thank you.
That was really interesting. Thank you for that wonderful talk. And thank you so much for keeping on time. I know that it's not easy to give your research review in such a short amount. I have a question. It seemed that over the multiple days where you had the color codes of all the different behavioral activity, pup approach maintained. So it's as if they're not learning the futility of their approach, so that they keep trying, like the effort persists. Is that true?
Right. So yeah, that's one way of interpreting this. We classify pup approach as when the adult actually directs their body and their movement, their snout and their facial areas towards the pup. So that's what counts as pup approach, versus search is like a random movement behavior. So we think they are interacting with them. We can see a little bit of whisker and interactions there. But so we think it's actually physical tactile interactions, which doesn't result in this. So that's why we think tactile perception is very important for this process, and that the het animals are somehow being aversive to this condition because of that pup approach phenotype. So they keep going back, but they don't actually finish it because maybe they find the pups aversive or something like that at this point. And actually, Dana would tell you a little bit more about this in the next step.
Okay, that's really interesting. Okay, great, thank you. Yeah, that's perfect timing. If we can, if Dana, we can switch to you and you can share your screen. Thanks again, Keerthi. Great, yeah, we're